Welcome, everyone. Thank you for joining us for today's webinar, Cannabis as an Anti-Cancer Treatment, the Scientific and Human Evidence. It is presented by Justin Kander, Research and Development Coordinator at the Oil Plant. My name is Judy O'Rourke, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Justin Kander. I will now turn the presentation over to him.
Hello, thank you so much for having me. It's an honor to present here, and I'm excited to get started in sharing this very important information. So jumping right in, how does cannabis fight cancer? Most of the research with cells in animals has focused on the two primary cannabinoids in cannabis, tetrahydrocannabinol and cannabidiol. The studies have identified four primary ways that these and other cannabinoids can fight cancer. The most direct method is the induction of apoptosis, or programmed cell death. This is where the cannabinoids bind to either the CB1 or CB2 receptors on the surface of cancer cells and trigger this programmed cell death where the cells break down into these little apoptotic bodies for neat digestion. In addition, the receptor activation can also inhibit the proliferation of cancer cells, stop the formation of blood vessels to solid tumors, which is a key step in their growth, and prevent the metastasis of cancer cells to other organs, which is also a key step in the progression of cancer. Most of the evidence demonstrating these effects has been conducted preclinically, with cell lines derived from humans and sometimes animals, as well as animal studies where the cancers are implanted in animals and the tumors are allowed to grow. The next level is anecdotal evidence, which is what most of the human evidence is found in. These are human stories that have been self-reported or reported in the media, where people who have used high doses of cannabis extracts are achieving remission. While most of it is anecdotal, there actually are a couple case reports that are formally published in medical journals that point to these anti-cancer effects as well. Finally, we desperately need clinical trials, placebo-controlled and double-blinded, to further demonstrate these effects. And while we do need more, there is one great trial that I'll talk about at the end, which backs up everything else. The preclinical evidence is quite remarkable in itself. Dozens of studies have shown how both THC and CBD can kill numerous types of human cancer cells and shrink tumors in animals. So this is a pretty complete but not all-inclusive list of the various forms of cancers that have been shown to respond in these preclinical studies. So this includes glioma brain tumors, various forms of breast cancer, leukemia, thyroid cancer, multiple myeloma. It really is remarkable just how many forms of cancer have been shown in these studies to respond. And I'm going to go over just a couple of these studies to show how they're done. So first, this is THC against liver cancer, specifically the hepatocellular carcinoma form. So the researchers found that THC induced programmed cell death in two different cell lines, and that mice who were implanted with these cancer cells and allowed to have the tumors grown have lower tumor growth when administered with cannabinoids. So as you can see with the controls, the tumors are much bigger than the cannabinoid treated group. In this case, the researchers both used the natural plant-based phytocannabinoid THC, as well as a synthetic cannabinoid known as JWH, or one of the JWH family members, which is a synthetic lab-made cannabinoid that activates cannabinoid receptors. And researchers do this to identify the mechanisms by which cannabinoids work, as THC has targets beyond the cannabinoid receptors, but this helps show that those receptors are indeed critical to this anti-cancer effect. CBD is also potent. It's been shown to kill 2 types of glioma cells, and administration of 0.5 milligram a day of CBD to mice reduced tumor growth pretty significantly. And while both THC and CBD have been shown to work on their own in these studies, the real power appears to occur when they're combined. So this is a study that looked at the synergy between THC and CBD for a form of glioma brain tumors. And the black bar on the very left is the untreated cancer cell. So at that point, the cell viability is at 100%. It's completely fine. With the THC and the CBD alone, the red and green arrows, there's a significant decrease in the viability. But when they're combined, the effect is much greater than either of them alone or additively, showing that they work synergistically in a greater than additive manner to inhibit this cancer. While THC and CBD have certainly gotten the most focus, all the major phytocannabinoids have been shown to possess anti-cancer properties, including the raw, unheated cannabinoids like THCA. This is a really great chart that just provides a good overview of how the different cannabinoids are shown to affect 8 different types of cancer cells. So going along, you have breast cancer, brain, prostate, thyroid, colorectal, breast, leukemia, and gastric cancers. And it looks a little complicated, but it's not too bad. Every number in these boxes represents the IC50 value, which is the concentration required to inhibit proliferation of a cancer cell by 50% compared to the control. So in this case, with the MCF7 breast cancer cells, it took 4.2 micromoles, which is about 4.5 milligrams of THC, to inhibit this form of cancer by 50%. Going below, you can see in this case the THCA and CBD were more potent, as it required less of these compounds to achieve that 50% inhibition. So it's basically the point of this is just to show how the various phytocannabinoids and the actual whole plant extracts, which are at the bottom, the THC and CBD rich extracts, work against multiple forms of cancer. There are also a couple of unique anti-cancer effects beyond those 4 that are pretty well characterized. One of the most difficult parts of treating gliomas and certain other tumors is the presence of stem cells in these tumors that are resistant to chemotherapy and radiation. One study that found that CBD could stimulate the differentiation of these glioma stem-like cells into more developed cells and prime them to respond to conventional treatments. And the researchers did indeed find that the CBD synergized with the chemotherapeutic agent carmustine to increase apoptosis in these cells, where the agent alone was not effective, pointing to the very powerful synergy that can exist between conventional and cannabinoid therapy. Immunotherapy has also been getting a lot of attention, and there's some evidence that both CBD and to some extent THC can help the immune system fight cancer. A study tested both cannabinoids on lung cancer cells and found it increased a protein called ICAM1, which is intercellular adhesion molecule 1, on the lung cancer cells. It kind of does what it sounds like and helps the cells adhere to special anti-tumor white blood cells that allows them to be destroyed. And the researchers found that this cannabinoid-induced increase in that protein was responsible for enhanced tumor cell elimination by the immune system. This is all pretty good information, but there are a lot of natural compounds that are capable of many of these things. And what really sets cannabinoids apart is the anti-cancer effects of our own endocannabinoid system. Endocannabinoids are the cannabinoid compounds produced in our own bodies that act in similar ways to the phytocannabinoids produced in the cannabis plant. And these compounds, primarily anandamide, have been shown to induce apoptosis through similar mechanisms as THC and CBD. So this is a few of the cancer cell lines that have been shown to respond to anandamide, and even indirectly increasing the levels of anandamide and the other primary endocannabinoid, 2-EG, is linked to the prevention of colon cancer in animal studies. So when you inhibit the degradation, you indirectly raise those levels, and that has benefits. With the strong science, it's not surprising that there's a lot of great human evidence to back this up. And I'm just going to go over a little bit here, but there's so much more out there. One of the most well-known case studies was published in November 2013 and described a 14-year-old female with terminal acute lymphoblastic leukemia who had not responded to chemotherapy and radiation. She used cannabis oil as a last resort, and doctors tracked the leukemic cancer cell count as she was using the cannabis. And as the chart shows, there was a dramatic decrease in the leukemic blast cell count while using the cannabinoids. While this chart happens to say hemp oil, this was made back in a time when that often referred to THC-rich cannabis oil. Now it's far more associated with CBD-rich oil, but this does indeed refer to a THC-rich treatment. Unfortunately, this particular patient did pass away as a result of a gastrointestinal perforation from the previous conventional treatment. However, the doctors remarked how the therapy had an increase in the patient's vitality, no toxic side effects, and had been able to control the blast cell counts. So if she'd been able to get the cannabis sooner, potentially with the chemotherapy, things may have worked out better. A more recent article focused on a 44-year-old man who was using vaporized and topical cannabis for a squamous cell carcinoma in his cheek. He was initially just using vaporized cannabis to deal with the pain of this cancer, but as it continued to grow, the vaporization became impractical, and he began applying a cannabis-infused sunflower oil on it, which was of a relatively low potency for topical cancer treatment. However, despite this low potency, there was a reversal of the tumor's growth after the topical had started. As you can see from the tumor size column, it was growing quite rapidly while the man was only vaporizing cannabis. However, once he started applying the topical oil, it actually began to shrink, which is extremely rare with a fast-growing cancer like that, and really only explainable by the potential anti-tumor properties. Unfortunately, given that he was using a low concentration oil and apparently not ingesting very much internally, he passed away from apparent metastasis. However, the case reporter's author, Dr. Vincent Maida, remarked how after a few weeks of the topical application, the malignant wound had started to regress, and this outcome suggested the topical medical cannabis may promote anti-neoplastic activity as per the findings of Casanova et al. That study he's referring to is one that used actually synthetic cannabinoids, including one of the JWH family members that the earlier liver cancer study used to show how cannabinoid receptor activation leads to the death of skin tumor cells. Most of the human evidence has been reported in the media or by people themselves through YouTube or Facebook. And it truly is remarkable how many stories there are, and I'll just go over a couple. One recent story focused on a woman named Dee Manny who had been diagnosed with triple-negative breast cancer in March 2017. She made the personal decision to try THC-rich cannabis oil as the sole therapy, which she obtained through a local supplier. She was taking 1 drop in a capsule every night before bed, and by August 2017, doctors determined she was cancer-free. And as this scan results show, it was quite a remarkable effect. And just to briefly go through a few other human cases, first we have Lynn Cameron, who was diagnosed with a brain tumor and given just 6 to 18 months to live, even with conventional treatment. She combined the oil with that treatment and achieved full remission within 6 MRI scans and is still doing well to this day. Augusta Fleming had been diagnosed with stage 3 lymphoma and underwent the full course of chemotherapy, which proved effective in her case. As a last resort, she moved to Colorado to take large doses of THC-rich cannabis oil, and within 3 months, a scan showed no evidence of disease. Follow-up a year later also showed continued inactivity. Finally, we have Gary Hill, who was diagnosed with lung cancer and given 6 to 12 months to live. At his age, he decided he didn't want to potentially endure chemotherapy and tried a legal form of CBD oil. Within 2 months, a CT scan revealed a near total resolution of his left lower lung mass, and most of his lymph nodes were normal size. And as of the publishing of that article, he was in remission. And what really gives even more credence to all of these cases is the fact that there is placebo-controlled evidence of an anti-cancer or a placebo-controlled trial showing anti-cancer evidence in humans. This was published in February 2017 by GW Pharmaceuticals. This was the results from their phase 2 placebo-controlled trial using a THC-CBD mix, their Sativex blend specifically for glioblastoma. So 21 patients were given either the THC-CBD blend and chemotherapy or the placebo and chemotherapy. While the study itself did not specify the exact dose, a physician from another conference who looked into it determined that it was likely only a maximum of 62 milligrams per day total cannabinoids that these patients were getting, which is very low for potentially achieving an anti-cancer effect, making these results even more impressive. The median survival for the cannabinoid group was about 6 months longer than the placebo group. And for a cancer like glioblastoma, where even with treatment, the 2-year survival is only 30%, a 6-month extension is very significant. And as the principal investigator, Professor Susan Short, remarked, indeed, the pharmacology of CBD and THC appears distinct from the existing medications and may offer a unique and possibly synergistic option for future glioma treatment. So overall, the evidence is quite strong for the use of cannabis for cancer. Both endocannabinoids and phytocannabinoids have been shown to affect many types of cancer cells through similar mechanisms, primarily the activation of the CB1 and CB2 receptors. However, other receptors often come into play. Some of these effects have been observed in humans through the case studies. And I do want to quickly discuss epilepsy because this really bolsters the chance that the preclinical evidence for cancer would also translate to humans. So it's very well known now that CBD is, and it's actually completely proven through GW's placebo-controlled trials, that CBD can reduce seizures in patients with Dravet syndrome. However, the preclinical evidence focuses on CBD for largely for chemical-driven seizures, which are far less complex than the genetically driven seizures that patients are experiencing with Dravet syndrome. And so if the preclinical evidence is translating so well to humans for that, it does at least make it somewhat more likely that other radical preclinical results, like for cancer, would translate to humans. And I do have a book at freecannabiscancerbook.com that has over 100 human cases and several animal cases, dogs and cats, along with extensive analysis of the preclinical evidence. And when all of this is examined together, it's very clear that cannabis can absolutely have an anti-cancer effect in humans and certain animals. However, we desperately need more research to characterize the extent and limits of efficacy. There's no doubt that many people don't respond to high doses of cannabis for their cancers for one reason or another. For example, if the tumors don't express high levels of cannabinoid receptors. That's why we desperately need more research, and anybody who uses cannabis for cancer should absolutely have medical oversight and keep close track of the cancer's progress to see if it's working. Nonetheless, while we have so much more left to learn, what the current evidence more than justifies the access of cannabis for patients who want it. And as time goes on, further research will help us get even better results. Thank you so much, and enjoy the rest of the conference.
Thank you, Justin, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through June 28th, 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.